Bismillahirrahmanirrahim. Assalamu alaikum, dear all students. This is the lecture about the next step in the histopathology laboratory and histotechniques, how tissue sectioning technique is performed. So, in this lecture we will learn about the requirement of tissue sectioning, the different instruments which is used in the tissue sectioning, that is the water bath, and the related instruments will be discussed in this lecture. So, the requirements of the tissue sectioning technique include the instruments and the other related materials for the tissue sectioning technique is a microtome with the blade. Second is the water bath. The third one is the paraffin block with the embedded tissue to cut. The ice tray is used for this tissue sectioning technique, and the forceps, which are blunt, is used. The brush used for this technique is the camel brush, and then the slide racks with slides. This is the water bath or the flotation chamber. Here we can see in this image, this contain a warm water with round about 30 to 37 degrees centigrade. This is the thermostat which is adjusted with this knob, the on off button and the heat button. This type of water bath or the flotation chamber is used to float the tissue after cutting. The temperature of this water bath should be 10 degrees centigrade below the melting point, that is above 30 degrees centigrade or from 40 to 50 degrees centigrade, so that the paraffin wax do not melt. Therefore, their temperature is kept lower than the melting point of that embedding medium. And this water bath prevents the formation of air bubbles during the tissue when they are cut, and also it should be taken in notice that no bubble formation should be found during water bath. So, the forceps and the camel brush, this is the blunt, this is the camel brush which is used for the tissue sectioning technique, and here are the forceps, these are the blunt forceps which is used for the tissue sectioning technique. During that, the blunt forceps helps to handle the floating tissue section, whereas the camel brush is used to clean the blade after the tissue sections are prepared and after the ribbon is prepared. So, this is the usage of this brush and forceps. The slide racks, these are used to keep slide in them, and these slides can be already labeled with the diamond pencil or on the frosted side by the lead pencil. So, these slides can be marked after lifting the tissue section. Here the slides in which the frosted side is here, the two different types of the slide racks. The adhesives which is used during this is, but it is not used in routine sectioning and routine staining. Some tissues need adhesive material for this tissue sectioning technique. For example, the brain sections, the decalcified bones, or using strong alkali for those sections for which we use strong alkali at the time of the staining. The most commonly used adhesive include the mare's egg albumin and glycerol. The next one is the poly-L-lysine and the 3-amino-propyl triethoxylene and the permanent positively charged slides. These are the 4 different types of adhesives used for the 3 different situations. These are the steps of the tissue sectioning. First, the trimming of the tissue in which the extra wax is removed from the tissue so that the actual tissue and the main tissue it get visible and it is prepared for a thin ribbon sections. Then after trimming of the blocks is placed by ice so that it get cooled, because when it comes in contact with the blade then these blocks get heated and there may be chances of the tissue distortion when a large amount of heat is produced. After cooling the block, then the small sections of the tissue is prepared and the ribbons is prepared. Then the ribbons is moved to this flotation chamber. This flotation chamber removes the wrinkles from the ribbons and the ribbons get straightened. After that, these are picked with the dry slides and they are kept for the drying. The trimming of the tissue. This is required to expose the tissue piece within the paraffin wax for cutting. And for this purpose, the block is fixed in the clamp of the microtome and the extra wax is removed, is cut to expose the tissue. It should be noted that the tissue should not be overcut because this may produce the artifactual changes. Like we can see, this is the block in which the tissue is present here in the center and it is covered with the extra wax. So the extra wax is removed by trimming, but here are certain cracks which are produced by the overcutting of the tissue. So these artificial changes comes during the overcutting or during the over trimming of the tissue. The next step is the cooling of the block. After the initial trimming of the extra wax, the block is then kept for cooling for 15 to 20 minutes so that the temperature gets normal to the room temperature. This is the image which shows the good ribbon in which the continuous section of the same tissue is prepared. The cooling of the block helps to maintain the same consistency of the paraffin wax and the tissue, and this helps us in easy cutting. How proper cutting can be done with the help of the microtome? First, the block is fixed in the holder. Then the cutting surface of the block is placed parallel to the knife. Just we can see in this angle, in this image. The angle of the clearance should be only 2 to 5 degrees centigrade to have a good section. Then after placing in a good angle, cut the tissue by gentle, smooth and slow stroke into ribbon like section, just we discussed and we see in the last picture. The tip of the ribbon is held by forceps and the end part of the ribbon is removed from the knife with the edge of the brush. Here, this is the first tip of the ribbon, and this is placed, and this is taken with the help of this blunt forceps, and the end is here, and the camel brush helps this ribbon, and the end is picked with the help of this camel brush. If you are struggling to get a flat section, then the tip for this is that first gently warm the cutting surface by a warm water. So you will get the thin flat section. When the tissue is placed in the flotation chamber, the ribbon of the tissue is floated in water to make it flat and to remove all the wrinkles that is produced during the ribbon formation and during cutting of the section. The individual and each section is separated with the help of the forceps, like we can see here are the multiple sections, and with the help of this forceps each section is separated and each section is taken with individual slides. It is needed to maintain the temperature of the water bath below the melting point of the wax, so that they get wrinkle free. If the temperature varies in the water bath, the air bubbles may be formed that may rupture the tissue. So this is the point to maintain the temperature of the water bath. If there is an issue with the water, so add a few drops of alcohol or a few drops of detergent in the water. This will help the reduction in the surface tension of the water, and the tissue will float smoothly in this water. Picking up the tissue, how the tissue will we pick up from this flotation chamber. So place slides vertically within the water bath in front of the tissue. Then when the tissue is touched, withdraw vertically. After that, this pickup process must be gentle and it must be smooth. Clean the water bath immediately after each block, so that the remnants, the pieces of the previous tissue, it do not get on the next tissue, and that is not intermixed with the previous tissue. Now when the sections are taken up with the slides, then these are dried up. The slide containing the picked up section is kept on a slide rack, and then they are kept in the hot oven to get dry. The temperature of the oven, it should be slightly more than the melting point of the paraffin, so that the paraffin melts and only the tissue is left on the slide. So that was all about the tissue sectioning technique, that how a tissue is sectioned with the help of microtome, then how the ribbon is prepared, if there is any difficulty in the ribbon preparation, how some tips which will helps in the ribbon preparation and which will helps in maintaining a wrinkle free tissue sections for picking up on the slides. Thank you. Assalamu alaikum.